Hi everyone, my name is Edoardo Porto and I work as a neurosurgery resident and research fellow at the Department of Neurosurgery of the Istituto Neurologico Carlo Besta in Milan, Italy. Today I'm going to present an abstract entitled A3D Ultra High Frequency Ultrasound Atlas of the Isolated Guinea Pig Brain Preparation, a new tool for focused ultrasound preclinical research. To start, I have no conflict of interest in relation to this presentation. The isolated guinea pig brain preparation is an invaluable resource for basic neuroscience. This anatomic model has been applied in several contexts, starting from the in vitro analysis of epilepsy pathophysiology. Recently, and surprisingly, it has also been used to gauge the induction of blood-brain barrier opening by means of low-intensity pulsed ultrasound waves and intravascular microbubbles. With this background in mind, the aim of our project was to perform a high-frequency ultrasound scan of an isolated guinea pig brain preparation and develop a free-accessible ultrasound-based comparative histological and 3D atlas labeled with key anatomical landmarks as a tool to drive future preclinical neuroscientific research. In this project, we use 3 Guinea pig brain and the Vevo station and the Vevo 3100 scanner, which is a ultra high frequency ultrasound scanner with the relative probe, which can be placed over our specimen and automatically slide over the isolated Guinea pig brain in a preset speed. As we can see, we collected in this video a B-mode imaging, which is magnified in this slide. This machine allows the high spatial and temporal resolution of the images acquired. And seeing from this consideration, we were able to compare these images with their relative quality with the gold standard imaging for the guinea pig brain, which so far is the histological coronal stain section of the guinea pig brain. So we basically selected 6 histological images readily available online from the craniocaudal axis on coronal sections, and we coupled them with some B-mode images that we acquired in sequential scanning. Of course, there is a limitation in this matching because when you use an isolated guinea pig brain, the brain lays dorsally on the pad for ultrasonography scannings, and this affects somehow the shape of the brain, which was not exactly similar to the shape of the section of the histological section. Nevertheless, these images, both histological and ultrasonography images, were capable to show some anatomical structures which were labeled in this project based on the available literature, which unfortunately is based just in hand-drawn atlases of the brainstem and the guinea pig supratentorial structures. So we were able to identify some cortical, subcortical structures, as well as structures of the basal ganglia. We identified 32 structures in these 6 couples of images in both the histological and ultrasound images. And among these structures, the only one which was possible to be recognized in all the images was the corpus callosum. Using the software VevoLab from the same manufacturer of Vevo 3100, we were able to reconstruct a 3D rendering of the guinea pig brain isolated preparation. And this technique showed a high resolution to identify the surface structure of the isolated guinea pig brain. For example, in the right side of the screen, we can see very clearly the olfactory bulb, while on the left side of the screen, caudally, we can see the ventral structures of the brainstem. In particular, the pyramids are very clear in this image. As a proof of concept in this study, we also attempted the contrast-enhanced imaging of the isolated guinea pig brain, and we were able, on the left side of the screen, to enhance the terminal branches of the arterial vessels which were quite visible in the cortex and also into the subcortical structure, as well in the basal ganglia. On the right side of the screen, we also applied another technique provided by this machine, which is the maximum intensity persistence technique, with even enhanced the capability to identify very tiny terminal arterial branches. Also, we tested the possibility of seeing the washing, and then after the performance of a burst shot, the retesting of the washing in, which is visible just in these moments. The contrast was perfused constantly through the use of a peristaltic pump in this study. In conclusion, the ultra-high frequency ultrasound revealed to be a valuable tool for guinea pig brain imaging, allowing the identification of anatomical structures as compared to the gold standard histological images. The presented comparative atlas can reveal to be an accessible and readily searchable resource for preclinical neuroscientific research in fields such, for example, neuro-oncology or movement disorder. Thank you very much for your attention.